So I'm going to start my tutorial now, and actually I've broadened the topic a little bit beyond just imaging to biomedical sciences in general, to have a little bit more breadth, which I think may be of interest to the participants. Okay, so before I start, let me say that most of what I'm going to say is in a book that I published recently with Cambridge University Press, which has both some of the history and some of the theory behind deep learning, and also applications in the natural sciences, which is what I've been focusing on, in particular application in physics, in chemistry, and biomedicine, which is the topic of our tutorial. So the one very important point I want to make about deep learning and biomedicine is that it's really a two-way, freeway between these two areas of science, in the sense that, of course, we are using deep learning in applications to biomedical data. And this is a very active area of research. There are applications of all kinds, at all different scales, from the atomic, from the molecular level, all the way to the imaging clinical level, as well as the population level. So it's a very, very broad area that we're covering today. But in the other direction, something that perhaps is not appreciated as well, is that a lot of the inspiration in deep learning comes actually from biology. And the obvious one is, of course, the origin of deep learning and AI in neuroscience, in trying to understand the brain. But you will see throughout my talk that even when you go down at the more technical level and you look at things like recursive neural networks, or graph neural networks, a lot of those ideas were actually inspired by were developed while working on problems in the biomedical sciences, such as protein structure prediction, for instance. All right, so this is the roadmap for the tutorial. I'm going to start with a little bit of historical background, showing how key ideas in biology, in neuroscience in particular, have provided the foundation for a lot of the things we do today in deep learning. So let's start with the historical background. The beginning of deep learning, I would say, is shallow learning. The difference between deep and shallow is not whether there are 3 layers or 5 layers, etc. The correct definition is that shallow learning is when you have no hidden layer, no hidden units. All the units see the inputs and the output. They are in contact with the outside world, if you want. And so the beginning of shallow learning is linear shallow learning. You can do also non-linear shallow learning, logistic regression, for instance. But linear shallow learning, of course, was developed by Gauss and Legendre at the end of the 1700s. And everybody knows what this is, of course. It's drawing the red line that is going through the blue points. But if you think deeply about what is in this picture, you will realize that most of the questions we address in deep learning are already contained in this picture, in the sense that you have data, you have a class of models, you have to define what does it mean to be the best model, so least square in this case. And then you can use the model for prediction. How do you use the model for prediction? And so on and so forth. So a cynical person could say that deep learning is pretty much linear regression on steroid from a certain point of view. And the big difference, there are 2 big differences, though, between this picture and what we have in deep learning. The first one, of course, is that there is a mathematical equation for the red line. Right, when you do when you do linear regression with trying to minimize the least square error, there is a mathematical solution to the problem that tells you immediately what are the slope and y-intercept of the line, or more generally of a linear hyperplane. And of course, in deep learning, we don't have that. You cannot solve mathematically for the best neural network if you want. So you're going to need some kind of approximation, progressive approximation of the solution, which is of course done with stochastic gradient descent. So that's the first big difference. It's a little bit of an outlier that in linear regression you can solve the equations. The second big difference is that in linear regression you can see things with your eyes, at least in 2 dimensions, maybe even 3. Beyond that, you cannot see it with your eyes, but you still have some intuition of how the hyperplane looks like in high-dimensional spaces. Although I'm not sure that intuition is very good. But in any case, in deep learning, we're going to use highly nonlinear surfaces or manifold, and therefore we cannot see things with our eyes. And that's the root, the origin for the feeling that deep learning is sort of a black box form of learning, and we don't really know what the neural networks are doing, etc., etc. I will point out to you that this is actually not too surprising, and the beginning of that is also visible in this in this picture, because if I give you the red line, which is the neural network when you go through deep learning, and I ask you to recover the points, the blue points, you cannot do that. The red line it contains a compressed representation of the blue point, essentially the statistics of the blue point, the mean and the covariance of the blue points. But it's a lossy process. When you do learning, you're basically taking the information in the data and you're pushing it into the weights in a lossy way. You cannot go backwards. And so it's not very surprising from this point of view that you're going to find that deep learning is sort of opaque or not very easy to interpret, at least on the surface. So nonlinear shallow learning is logistic regression, so one could go to that, but more broadly, I'm going to go directly to deep learning, and the beginning of deep learning is definitely around 1950. And so I'm going to review what was known about the brain around 1950 very briefly, to give you a sense of what was the state of the art at the time and what were the basic ideas that were used to develop deep learning. So, of course, people were already thinking about the brain, and a lot was known about the brain. In the upper left, you have Charles Darwin. So people knew that the brain is a product of evolution. So it's very messy because evolution operates by tinkering. They knew that the brain had specialized areas. People like Wernicke, Charcot had discovered brain areas, for instance, involved in speech. So that when you have damage in those areas, you have trouble with language, with speaking or with understanding language. Ramon y Cajal on the upper right had done a lot of anatomical studies. So they knew about neurons, about pyramidal cells, Purkinje cells, and so on and so forth. Sherrington here in the middle on the left had developed, had introduced the notion of synapse. So they knew that there were points of contacts between the axons of neurons and the dendritic trees of neurons. And in the 40s, late 30s, and continuing in the 40s, all the way in the 50s, Hodgkin, Huxley, and others have started studying how neurons communicate with each other with very tiny electrical signals. So people knew that neurons send signals electrically to each other, that these signals travel along the axons and so forth. On the theory side, by 1950, you had Gödel, you had, of course, Turing, you had Shannon. All the basic theory of information was in place due to the work of Shannon. Of course, the first computer architecture were being developed, von Neumann. But then you had these two gentlemen at the bottom here, McCulloch and Pitts, who wrote an influential paper, which perhaps is the origin, the real origin of deep learning, where in this paper they introduced the notion of artificial neuron, what we use now in deep learning. which I'm going to describe in a second. Here it is. So of course, you know, this model, very simplified view of how a neuron may operate, where a neuron is viewed as a system that operates in two steps. First, it takes a dot product or a weighted average of the inputs. The x1, xn are the inputs. W, wn here are the weights. So you first take the weighted average, and then you pass it through an activation or a transfer function, which if it is the identity, of course, you get a linear neuron, but more generally, we use nonlinear activation functions in deep learning. At the time, they started thinking about sigmoidal or threshold functions like the heaviside or the sine function for threshold functions. For a sigmoidal function, you have the logistic function or tanh. Of course, today we have also other activation functions, especially piecewise linear activation functions such as ReLU and so forth. And this is how the neuron produces the output. Now, I described the way the neuron operates, but really there is something else which is very fundamental in this picture that is not always appreciated as well, is that it's not only a computing device, but it's also a storage device. So you see that storage and computing are intertwined, unlike what we have in a digital computer, and storage is done in these weights here. This is where you store the data. This is where the blue dots of the linear regression end up being projected in some sense to the learning process. And this is where the program is stored, because a neural network can also be viewed essentially as a computer program, and the information about the program is stored in this way. And this way of storing information is very, very different from the way you store information on the digital computer. And that's absolutely fundamental to think about that, and to think about how we store information in the brain. So even in the 1950s, of course, you could start asking the question, you know, think about your telephone number or your name. It is stored somewhere in the brain, but where is it? The true answer is that we still don't know today where do we store our telephone number. But anyway, you could start already in the 1950s to make the hypothesis that it's probably stored in the connections between the neuron in the synapses, because this is where you can rapidly retrieve the information. If I ask you your phone number in less than half a second, you can recall the phone number, and spit it out. You could not do that if the information was contained, let's say, in the nucleus of the neurons, in the DNA of the neurons, etc. There may be a trace of the information in the DNA through epigenetics, etc. That's a different story. But for the rapid recall, it is probably likely that it's inside the synapses. And this is just a picture so that you see how small synapses are. And so the question is, you can try to think about this problem. How do the brain may be storing information in synapses? Now, synapses are very small, so you're not used to think about that sort of length scale. They are on the order of 10 to the -7 meters. So what I'm going to do to make things more palatable to you is multiply everything by 10 to the 6. Rescale everything by 10 to the 6. And so a synapse, the diameter of synapse becomes 10 to the -1. That's about 10 centimeters. So it's the size of your fist. So imagine now that you are in a room, you are a synapse, it's the size of the fist. Imagine you're trying to learn how to play tennis or how to play the violin, for instance. And I ask you, well, where is the tennis racket? Well, you are in Oslo, or actually most of you, I mean, those who are at the conference are far away from Oslo, but the tennis racket is something like 8,000 kilometers away on that scale. So ask yourself, how is this fist that you have in a room able to decide whether it should strengthen itself or weaken itself, for instance, or change something about its chemical composition so that you play better tennis when the tennis racket is 1,000 kilometers away? That's really the center of deep learning. It's the fundamental mystery, where you have something that is a synapse synaptic weight, if you want, which is very small, very far away from the inputs and outputs, 1,000 kilometers away. It doesn't seize the inputs, the outputs. It doesn't know anything about tennis. It doesn't know anything about Newton laws of mechanics or anything like that. It doesn't have a cell phone, cannot Google the answer. How does it decide whether to go up or down? That's the fundamental question behind deep learning. And one of the first person who thought about this problem is Donald Hebb, who wrote a book at the time. And in this book, he wrote the famous sentence that is here in bold, but which can basically, paraphrased. Usually it is paraphrased in the form, neurons that fire together, wire together. But it's the idea of correlation learning, right? If you have a synapse connecting neuron J to neuron I, if the neural activity of I and J is correlated in some sense, then the synapse should strengthen itself proportionally to that correlation. So mathematically, very often we write this as OI times OJ, where OI and OJ are the outputs of the neurons. And this is a nice idea because it is physically plausible, right? The rule for changing the weight between I and J, neuron I and neuron J, is really dependent on local variables. You don't have the 1,000 kilometers away problem. Everything is local, which is what it has to be that way in the brain, right? A synapse can only change itself on the basis of local information. So that's very nice. On the other hand, the problem is that it's not clear why, if you were to apply this rule to a large network with millions, billions, trillions of neurons, why would you get, intelligent behaviors just by applying this very simple, stupid-looking rule? In fact, nobody has been able to build anything really smart using Hebb's rule alone. But that was the beginning. And then Rosenblatt, a few years later, came up with the perceptron rule. So basically, if you study the perceptron rule very carefully, you will see that it is essentially gradient descent applied to a single McCulloch and Pitt neuron. So that was the discovery of Rosenblatt. And very importantly, in the 60s, these two gentlemen, Hubel and Wiesel, began studying the cat visual system. So they recorded from the first visual areas like V1 in anesthetized cats. And they made a famous discovery that in those areas, one of the things that you find is you find arrays of cells that have very particular responses. They can respond, for instance, to bars at a particular orientation and a particular location in the visual field. And you will find in these arrays neurons that respond, let's say, at bars that are oriented at 45 degrees at every location in the input field. And then you will have neurons that respond to bars at 90 degrees, every location in the input field, and so forth, and so on and so forth. And if you go on YouTube, I don't know if I can show it to you, there is a very nice video of the experiment of Hubel and Wiesel. Can you hear the sound?
Yes, we hear it.
That's okay. So I'm just gonna go very fast through the video. Let's see. The bar is not at the right orientation. You see the neuron fire. You can hear the firing of the neuron. They feed it through a microphone, of course. And when the bar is at the right location, right orientation, you hear the neuron. If you change the orientation or the stimulus, then it doesn't fire. So it's a very interesting, I'm not going to play the whole video, but I recommend watching it if you've not seen it. Okay. Oops, now my present, okay. And this is what inspired Fukushima to come up with the idea of what we call today convolutional neural networks. So in the 70s, he developed this idea of the neocognitron, what he used to call the neocognitron, but it's really a convolutional neural network, where he basically took inspired by the discovery of Hubel and Wiesel. The idea is that in computer vision tasks for recognizing objects, for instance, you're going to build the networks that have many layers. And in the early layers, you're going to have banks of filters or arrays of feature detectors, which are these, similar to Hubel and Wiesel, detecting bars at different orientations, for example. And the question is, how do you learn those connections, right? And Fukushima didn't use what was available at the time, and the only algorithm for a deep network was Hebb. So he thought, if you read his papers, that you could use Hebbian learning to adjust the connections in this architecture, right? Unfortunately, this has never worked. Nobody has been able to train a deep network by using Hebbian learning on ImageNet. And in fact, there is a reason for that. Today, we understand mathematically that this is impossible, right? Hebbian learning will never work in a deep feed-forward architecture for complex tasks like image recognition, biomedical imaging, and so on. The algorithm that works, of course, is called backpropagation. It's nothing else than stochastic gradient descent. It was derived multiple times by a number of people, but it's basically just a chain rule. So you just straight out of Newton and Leibniz, you can derive the backpropagation equation. But the way they look like is that if you have a deep weight, remember the story of the synapse when you're learning to play tennis, right? You have a deep weight here in this potentially gigantic network, very far from the outside world. And the way you're going to adjust it is incrementally by an amount that is described here that looks Hebbian because it's the product of two things. And one of the things is the output of the presynaptic neuron. So it's OJ. So that's very much like Hebb. But then the postsynaptic component is not OI. It's not the output of neuron I. It is something called BI, which is the backpropagated error, which is a signal that comes from the top where you compute the error, whatever. This is supervised learning, of course. So you have input, outputs, you have targets, you compute some error. Hopefully it's differentiable. Most of the errors we use are differentiable. And then you compute the error at the output layer, and then you propagate using the chain rule, this error all the way back to the weight Wij. And the propagation backwards is very similar to the propagation forward, except that it's entirely linear. There are no transfer functions. And you're basically using the same weights as in the forward direction. So in the reverse direction, you're going to have the transpose from a mathematical standpoint. And then in addition, every time you traverse a layer, you have to multiply by the derivatives of the transfer function, of the activation functions of the neurons in the forward pass. But that's the algorithm. It's very easy. It's a 3-line of chain rule, application of the chain rule to derive this equation. And this suddenly makes things work. And people, even in the 80s and early 90s, were able to train the first convolutional neural networks using this algorithm, backpropagation. The example that you're all familiar, probably all familiar with, is the NIST data set of Yann LeCun, where you can train in this way a convolutional neural network to recognize digits. But even in our own work, I think the first application to biomedical images was done by us to the problem of fingerprint recognition, where what you have here is what you would call today a Siamese deep neural network with two branches, one branch processing a fingerprint image on the right, a fingerprint image on the left, and it's in this case, it's a classification network which produces the probability that this is the same finger, the image of the same finger or not, right? And this was, again, developed in the late 80s, early 90s, and worked quite well. All right, so what has changed? So since things were already working in the 80s and 90s, what has really changed? One thing that has changed, of course, is the explosion in the data. I'm not going to spend much time on this, but just to give you a couple of key ideas. You look at other sciences like astronomy. You see here Galileo, who built one of the first telescopes with a magnification factor of 20. And today, things look like this. In fact, if you looked at the news in the past month or so, you saw that there is a new telescope that is going to replace the Hubble telescope called the James Webb Space Telescope, which is going to be at more than about 1 million miles away from Earth. It's an incredible piece of machinery. I should definitely change this slide. But anyway, the point is that Galileo could do everything with his eyes and with his brain. And when you scale things up to what you have today, you cannot do things with your eyes and with your brain anymore. You have instruments, complex instruments that are producing terabytes, sometimes petabytes of data. The LHC, Large Hadron Collider in Geneva, is capable of producing 1 petabyte of data per second. So much data that you cannot even store it, so you have to throw it. Most of it has to be thrown away very rapidly. But anyway, you get these very large amounts of data that have to be, first of all, stored in large databases or computer farms. And then, of course, you have the problem of interpreting the data, and that's where you're going to need statistical methods, in particular deep learning, to analyze this data. This is the center of the Large Hadron Collider, where you are colliding protons at speeds close to the speed of light, right? 1 petabyte of data per second. You see the same thing in biology. So invention of the first microscope with a magnification factor of 100. And in biology today, whether it's imaging or whether it's DNA sequencing, in this case, this is a Illumina sequencer that is capable of producing a human genome. Not this particular model, but the current models, a human genome that looks something like this in a couple of hours. This is actually the genome of the AIDS virus, which has 10,000 letters of DNA, and it fits in 1 slide. That's why I have it here. Of course, you see that it's very opaque and impossible for humans to read this very strange sequence of ACGTs, of DNA nucleotides. And of course, you're going to need algorithm, statistics, deep learning, etc., to help you make sense of this genome. Your genome looks actually exactly the same. It's a long string of ACGTs, but it's a little bit longer. It's like 3 billion letters instead of 10,000 letters. And then imaging, since the tutorial is centered on imaging, of course, you have all kinds of imaging in biomedicine, whether it's microscopy, whether it's X-ray, whether it's MRI. You have video when you do colonoscopy, endoscopies. I'll give you examples of what happens with videos. You can take images with your cell phone. You have a mole on your skin. You take a picture with your cell phone. You send it on the cloud. You would like to have algorithms and deep learning algorithms that analyze those images and send you some feedback, whether you should go see a doctor or not, and so on and so forth, right? So many different imaging modalities are used in medicine, different types of imaging, and at all different scales, from the microscopic all the way to the full organism and beyond. So we'll see how, and in all these cases, you can most of the time use convolutional neural networks in different ways. I'll show you how We do that in a series of examples that I will present. And by the way, most of what I'm going to show and what I'm saying is taken out of my personal experience, and I don't mean at all to imply that I'm the only person working on this in this area. There are many people, especially today, working on these problems. But I'll show you the kinds of things that my lab has been doing, because those are the one we know the best. And all right, so let me now say a few things about theoretical developments. I'm not going to spend a lot of time, except for one thing, which is what today people call graph neural networks, I think is the terminology used today. But I used to call them recursive neural networks. But the basic idea is this, is that you have two types of problems. You have problems where your data comes with inputs that have fixed size. So, fixed vectors, fixed matrices, etc. Imaging is in that class. Typically, you have images, they all have the same size. So you can view them as matrices if you want. So inputs of data that comes with a fixed format or fixed size. That's one class of applications containing imaging. But then you have also applications where the data comes with variable size. You have objects that have variable size, and very often they have some kind of graphical structure attached to them. So example of that are problems in chemistry where you have molecules. These are labeled, small labeled graph. Small molecules are represented by small labeled graph. That's an example. All the problems based on sequences, whether it's DNA sequences, RNA sequences, protein sequences, or language, NLP, natural language processing for that matter. You have sequences, so there is the graphical structure, right? The order of the letters is important. So you have a very simple left to right sequence of letters, if you want, in this sequence problem. But the length of the sequence is variable, whether it's NLP or whether it's a protein sequence. The length can vary quite a bit. And so when you have these problems, the question is, how do you apply neural network to these problems where you have both graphical structure and variable length? And for doing that, for developing these recursive neural networks, I think there are two main approaches that is good to keep in mind, which I call the inner approach and the outer approach. And I'm going to describe them to you in a second. So in the inner approach, you need to have a directed acyclic graph, which is a DAG. And basically, you're using neural networks to crawl the DAG, to move inside the graph from node to node. I'll show you exactly what I mean in a second. So that's what I call the inner approach, because you are inside the graph of your data. The outer approach is also called sometimes the convolutional approach. In this case, you're building your neural network in a direction that is perpendicular, perpendicular to where the graph of the graph of the data lies, right? So imagine the graph, a molecule is in a certain plane. You are building your neural network in a direction that is orthogonal to that plane. So let me show what I mean on simple examples. So imagine you have sequences, and you're trying to analyze those sequences or generate those sequences. If you're trying to build a generative model, whatever you're doing, you have to think about a model for those sequences. So what I've drawn here is an HMM, right? If you know something about graphical models, this is an HMM, which is a structure where you have a chain of hidden states, where at each time point, you're moving from a hidden state to the next hidden state to the next hidden state, right? You have in the graphical model formalism, you have the probability matrix that tells you the probability of each transition. And then when you are in a given hidden state at time t, you produce a symbol of the alphabet at time t. And so you have another stochastic matrix in an HMM that tells you what's the probability of producing, say, letter A when you are in this particular state. Okay, so that's the graphical model. It's a DAG, it's directed acyclic. And so when you transfer this in the deep learning world, all you're doing is to place neural networks on the edges of this graph. So it becomes something like this. You are in a hidden state at time t. There is a neural network that produces the hidden state at time t+1, and then a neural network that produces the hidden state at time t+2. Of course, you can do weight sharing, so you can reduce the number of parameters by reusing the same neural network at every times point to implement this transition between hidden states. And then you have a second neural network that takes care of the production. So as input, it's going to receive a vector representing the hidden state, and as output, it's going to produce one of the letters of your alphabet, or more generally, a softmax with the probability of your, say, 4 letters, a CGTs, if you're doing a DNA or whatever it is, 20 letters, if it's proteins, the 20 amino acids. And again, you can do weight sharing of the, you don't have to, but you can do weight sharing of the neural network at these different time steps. And so basically, you can refactor an HMM into 2 neural networks, one controlling the transition between the hidden states and one controlling the production of the symbols attached to each hidden states with weight sharing. Okay, and these 2 neural networks can be very big. They could have 100 layers for that matter, but conceptually, you just have these 2 neural networks that are implementing this sort of HMM idea. And of course, now you can take any kind of graphical model that you can think of, input, output HMM, bidirectional HMM, and so on and so forth. I'll show you more examples, and refactor them using neural networks. And because it is a DAG, there are no directed cycle, and that's the key thing you need in order for backpropagation to work. As soon as you don't have any directed cycle, you can just, if you have targets here at the output, you can just backpropagate through this entire structure through your 2 neural networks and train such a network. So that's the idea of the inner. You see the neural network are inside the edges, they are crawling the graph. In the outer case, you have your sequence at the bottom here, the blue dots, and you're building your neural networks in a direction orthogonal to the sequence if you want. You can, that's one way of looking at it. So you have first the blue neural networks, where a neural network may be looking at one point in the sequence and its neighbors. You could have a window of a certain length, and you do this at each position. You can do weight sharing. In this case, you have all the blue triangles represent the same neural network with the same weight applied at different positions in the sequence. The output is some hidden vector. And then you do it again using the green neural networks, the red neural networks, and so forth. You can have a stack. Each green neural network, for instance, could have 20 layers, right? And as you go up, the key point is that if you look at the hidden representation at a certain position here, position I, and you look at what size of the input is influencing this output here, the size of the window gets bigger and bigger. So the higher you go in the hierarchy, the bigger the input window that you're able to cover with your neural network. So you're able to integrate context that is far away from the position you're looking at, which is very important for natural language processing or protein problems, for instance, or DNA problems. All right, so let me show you rapidly how this works when you apply to proteins, for instance, a protein structure prediction problem, very famous problem, very difficult problem. It has many facets if you study it carefully, where you first want to predict the secondary structure. So starting from the primary sequence. So protein is a sequence over an alphabet with 20 letters corresponding to the 20 natural amino acid, which you see here on the right. The length of the protein sequence is very variable. The smallest length can be something like 30 amino acids, 30 letters. The longest one can be several thousands. An average, typical average would be maybe 500 letters in the human genome, something like that. And these sequences, they fold up in very complex 3D structures, which are important for their function. This is what determines what other molecules a protein binds to, whether it's, there are proteins that bind to DNA, they are called transcription factors, for instance. There are proteins that bind to other proteins, as in this drawing, you know, you have the red chain combining with the green chain, it's forming a protein complex. You have proteins that can bind to oxygen and carry oxygen in your blood. You know, hemoglobin or myoglobin is storing oxygen in your muscles, and so on. So the first level of prediction is prediction of the secondary structure, which is predicting where these structural motifs occur. The structural motifs are the alpha helix that you see here. There are several alpha helices in these proteins. So you want to know where are these alpha helices along the backbone of the protein. Another important structural motif is the beta sheet, where you have regions of the proteins that are called strands that are marked by arrows that are forming hydrogen bonds with each other. So you get many such arrows that are bonded to each other, and they form sort of a sheet called the beta sheets. And then what's in between is called the coil. So you see a coil, piece of coil here and in other places. And so secondary structure prediction at the coarse level is to predict are the helices, the coils, and the strands along the protein sequence. From there, that's called the secondary structure prediction. From there, you want to do ternary structure prediction, which is basically predicting the XYZ coordinates of the backbone of the protein. And you can also do full 3D structure prediction, where you predict not only the backbone connecting all the amino acids, but also the side chain, all these additional atoms that you see here on each one of the amino acids, which are sticking out of the backbone. So basically the XYZ coordinates of all the atoms of this complex structure. And then of course you can do also quaternary structure prediction, where you have different protein chains like the green and the red coming together, forming a complex, and you want to know how they are attached to each other. And so predicting XYZ coordinates of everything inside the complex. Okay. Now, if you think about applying a neural network to this problem, starting from the sequence and getting XYZ coordinates, that is going to be almost impossible to do, for the very simple reason that the problem has a translation rotation invariance. If I translate or rotate a protein structure, the structure stays the same, XYZ coordinates are going to change like crazy, right? So that is not going to work well with neural network. You would have to either define some kind of canonical reference frame and work just in that frame, reduce everything to the canonical reference frame, which is not natural. There is no canonical frame for these proteins, or you have to find a representation that is invariant to translation and rotation. And so this is what we and others have done. So this is the pipeline for 3D structure prediction, which is still used today, essentially in the current predictors, where you have the primary sequence. If you want, you can predict secondary structure or other things from the primary sequence. There is something called the solvent accessibility, basically predicting whether a given amino acid, a given letter is on the surface of the protein, so exposed to the water or to the solvent, or whether it's inside the core of the protein. Usually it's hydrophobic, doesn't like water, and you are in the core of the protein. So predicting whether you're on the surface or in the core is also another, classification problem you can work on. But the key step is this one, where you are predicting what is called the contact map or the distance map of the protein, which is a translation rotation invariant representation of the protein. And if you've never seen a contact map, just think that you're putting the protein sequence along the x-axis, you're putting it along the y-axis, and here it's basically a matrix of 0 and 1s, which is very sparse, and you put a 1 at position yj if and only if amino acid I and amino acid J are close to each other in 3D. Right? So you have I and J along the sequence, position 50 and position 200, right? There are 2 amino acids at those positions. When the protein fold, in some cases, they may end up being close to each other. There is a precise definition for closeness in terms of angstrom. I'm not going to get into the details. And it's a relatively rare event. Not all amino acids are close to each other. Most of them are far away from each other, but a few of them are. And that's what you get in this matrix. It's a symmetric matrix, of course, and it has ones on the diagonal because every amino acid is attached, covalently bonded to the next amino acid. And the big problem, of course, is to predict the corner, the lower left corner of that contact map, where you have very sparse contact, but they are very important. These are amino acids that are far away along the sequence, but close to each other in 3D. And that's the contact map, or you can work with the distance map, even better, where instead of a 0 or 1, you put the actual distance. So you put 7 angstrom, 12 angstrom, 50 angstrom in each entry of this matrix. And then from the contact map or the distance map, you can try to recover the 3D structure up to mirror images, because if you take the mirror image of the protein, the contact map stays the same. So there is a little bit of a degeneracy there, but it's not a big problem. So you can break things in those 3 steps. So I'm going to show you how you apply deep learning to the first 2 steps at least. The third one is, there are many techniques which are, AlphaFold has been using deep learning also for the 3rd step, but there are also techniques from distance geometry, for instance, to deal with the 3rd step. So first step, prediction of secondary structure, that's what we did. You build a little bit a graphical model, a little bit more complicated than an HMM, where you have inputs. You have 2 chains now, one backwards and one forward. So that's called the bidirectional model. And then you have outputs here. And so you see that the output at time t depends on the hidden state on the backward chain, the hidden state on the forward chain, and the input. The input can be a single amino acid or a window of amino acids or something a little bit more complicated that I'm not going to go over, but which is called the profile. But that's a detail on this level. And so again, now you want to refactor this input. This is called the bidirectional input-output HMM. And when you convert it to neural networks, you're going to get 3 neural networks. One that produces YT as a function of these 3 vectors. That's your output neural network. And then for the chain BT, you're going to have a neural network that computes BT, the vector BT, as a function of BT plus 1 and the input UT. And then for the forward chain, you're going to have a neural network that computes FT as a function of FT minus 1 and the input vector UT, right? So 3 neural networks. When you unfold the whole structure, you get this. So you see this is the forward neural network of the forward chain, passing its output to the next level, etc. This is the backward chain. This is the outputs, right? It's acyclic, so this whole graph is a feed-forward graph, very deep, but feed-forward graph. And you get targets here. If you have a database of protein structure where the secondary structure is known, so there are databases of proteins that have been solved by using X-ray methods. And so you can backpropagate through the structure and learn. And I'm not gonna spend time on a lot of details, but if you see, here's how the problem has evolved since the late 1970s. First approach by Chu and Fasman in 78. It's a rule-based approach, very simple, based on basic statistics, about 60% correct. Karen and Sinowski were the first to apply a neural network in the 80s to this problem. They used the convolutional approach, the outer approach, 64%. And then there were a sequence of improvements. We did this bidirectional stuff in 99, we were at 78%. And now today we are between 95% and 100% using these methods, but of course with much more data. Data has been growing exponentially. And so the secondary structure prediction is basically solved. There are reasons why you cannot get to 100%. There is a variety of reasons that are well known that I'm not going to get into. So then there is the question of the contact map. How can you do the contact map prediction? And one idea is to generalize what we did in 1 dimension to 2 dimensions using the inner approach. How does it look like? Well, now instead of chains, you're going to have to deal with planes. you're going to have an output plane, an input plane, and then 4 hidden planes, 4 lattices, where you're propagating information towards the 4 cardinal corners, northeast, northwest, etc. So if you look at the connection within one column, this is what you get. Your output at position ij is going to depend on the 4 hidden vectors in the 4 hidden planes right below it and on the input. And then if you go in the northeast plane, the vector at position ij is going to depend on the 2 neighbors in the lattice and on the input, right? So it's 2-dimensional generalization of the idea of bidirectional input-output HMM. If you understand this, now you know how to do it in 3 dimensions, right? You would have cubes, 1 output cubes, you have up to 8 hidden cubes and 1 input cube, right? If you write the equation, this is what it is. It can be refactored in terms of, in this case, 5 neural networks. The output neural networks that computes the output vector at position ij as a function of the input and the 4 hidden vectors in the 4 hidden planes. And then you have the 4 propagators that are propagating information in the hidden planes, right? The output, if it's a contact map, it's a 0, 1. So you probably have a logistic unit, right, that is trying to predict a 0, the probability of being a contact or not a contact. So you already know how this is going to look like. You can check that this is directed acyclic, again, back propagation, etc. So that's one method for trying to predict contact map. So that's the inner method. You can see how things propagate there. The other possibility is to use the outer approach. So that's the idea where you have the contact map. Imagine it a little bit like an image that you want to produce. So you start from a plane, and then you build your neural networks, possibly convolutional neural network as envisioned, on top of this contact map. You can stack them up as in the example where I was stacking neural network on top of the picture. You can have a lot of depth. And finally, you predict your contacts or your distances. And this is something we developed in 2012. This is very close to the basic idea of AlphaFold, that is the famous system produced by DeepMind, which works very well on protein structure prediction. But the core is to use this approach, the outer deep learning approach, for predicting the distances between the distance map, the distances between all the pairs of amino acids. All right, so let me now start with the applications. I will very quickly show you other applications besides proteins, and then I will move to imaging. So there are many applications, for instance, in drug discoveries, where you're dealing with small molecules. The space of small molecule is very large, much larger than the space we live in, because the space we live in has about 10 to the 22 stars, right? There is 10 to the 11 galaxies and about 10 to the 11 stars per galaxy. The space of small molecule that we know that have been identified on Earth or synthesized by chemists or found in biological system is on the order of 100 million different small molecules. But the space of virtual small molecules that you could make using the basic rules of chemistry is much larger. Estimates can be in the 10 to the 60 to 10 to the 100 range. It doesn't really matter. But so you have this, enormous space, and obviously computers should become the main tool of chemists in order to travel this space and to find molecules that are useful. For instance, drugs. Useful depends, of course, on the application you're looking at. So here is the molecule. So small molecules are defined as in organic chemistry. So these are molecules that are primarily made of nitrogen, carbon, oxygen, and hydrogen. Occasionally, you could have other atoms, sulfur, and so on. But those are the 4 main building blocks. And there is no hard threshold for what is a small molecule, but we could say, let's say something with less than 50 atoms is a small molecule. And so you have an object like this, and you would like to predict whether it's toxic or whether it binds to something or whether it's soluble in water and so on, right? Solubility is very important for drugs. And so the nice thing of molecule is that they can be represented in many ways. Here are at least 4 different representations. And so 4 different types of data that you can use for your deep learning. You can work on the graph directly using inner and outer approach, with some challenges that the graph is not acyclic. You know, if you have an aromatic ring, a benzene, you can have a cycle. So you have to deal with that problem. They are definitely variable size. So that's one way of representing a molecule. Another way is to use what is called smile strings. So you see here a string that represents this molecule. It's very easy to see how to map one to the other. This equal sign represents a double bond, for instance. You can view a molecule as a set of atoms. So you could give just the XYZ coordinates of all the atoms in the molecule to represent the molecule. That's another representation. The interesting challenge there is that there is no ordering, right? It's not really a list of atoms, it's just a bag of atoms. You can permute them, XYZ coordinates of the first atom, the 5th atom, and so on. It's a set of coordinates of atoms. So that's a challenge. There is another representation you see here as a vector of 0 and 1. That's called a fingerprint. It's basically, there is a 1 if a particular, say, chemical group is present in the molecule or not, otherwise it's a 0. And that's how you get these fingerprints. So the fingerprint is the only representation that consists of vectors of fixed length. So you could, in principle, use a feature-aware neural network on the fingerprints. But for all the other ones, you have to use graph neural networks or recursive methods, right? Because they vary in length, in size, and they have some kind of graphical structure. Very briefly, this is the one inner approach for molecule that we have developed to address the problem of not having a directed acyclic graph. What we do is we take a molecule, we replicate that has a certain number of atoms. In this case, it has 4, 6, 8, 9, whatever. So you replicate the molecule as many times as you have atoms, right? And in each replica, you choose a different atom as your sink, and you orient all the edges toward that sink. So now all the mini graphs, the little replicas, are acyclic, and now you can crawl them with a neural network that is going to take all the parents of the node and produce the vector of the child node, and then crawl the molecule until you reach the sink vector and produce the sink node and produce a vector in the sink node, right? And so we crawl all these molecules, we get all the vectors in the sinks, then we can add them together, for instance, and pass them through a final network that predicts whatever you're interested in, whether it's solubility, toxicity, and so on. There are almost an infinite list of possible properties that you may want to predict. Of course, you need a data set for training the system. But again, everything is, you can unfold it. There are no cycles, so you can backpropagate through the structure, and you can learn that. You can do it in the outer way. This is work that was presented at NIPS by Duveneau et al., where the molecule now, the graph of the molecule is in this plane at the bottom, and you're building neural networks on top. So at the first level, you could have a neural network that looks at an atom and its neighbors on the graph of the structure, and so on, and you stack them up and have the prediction at the top. So inner approach, outer approach for molecule. I gave you examples for proteins. You can do another interesting problem in biochemistry is predicting chemical reactions. I'm not going through that because we're a little bit running out of time, so I'm going to move rapidly to the imaging imaging type of problems. This is just an example of a problem, though at the genomic level, where you have the genome sequence here, and you want to predict certain things related to how these sequences are regulated. So you can use a bidirectional neural network, convolutional neural network for doing that. But what I want to do now is to show you applications to biomedical imaging, of which I have quite a few. But now we're back to the case of objects of fixed size images. And of course, we have convolutional neural networks from the Fukushima area, from the Hubble-Wiesel area, whiz-back propagation. Of course, today we have much more data and we have GPUs. Those are the two ingredients that have made a big difference, the computational power and the training sets, right? And so we can apply those to images of all kinds. And I'm going just to give you an example of such problems. So this is what you see here is a technology, new technology that is developed in a lab on my campus by Chris Hughes. And what you have there is a microfluidic device where you have little chambers, essentially, where you can grow blood vessels. So what you see in green here are blood vessels that are growing in these little chambers. And then you can, for instance, inject cancer cells and let the cell proliferate, and you can inject a drug, and then you can see whether the drug is shrinking the cancer cells or not, right? So you're going to see it on the next slide. The blue are the cancer cells, right? And you can do these various experiments, and you can do this in high throughput fashion, and you get these images under the microscope. And you don't want to have humans looking at these images and decide whether, you know, the cancer cell have expanded or shrunk due to the drug or anything like this, or whether the vasculature has changed from an image to another, whether it's normal or not, etc., etc. And so you can train a deep neural network to do that, to look at, compare images, for instance, and decide whether there's been a shrinkage of the cancer cells or changes in the vasculature, etc. There are different problems. In this particular example, you can see the ROC curve here. We get almost perfect prediction on these problems. I'm going to skip this one. This is another interesting project we worked on, which is looking at mammograms. So here you have X-rays of the breast. Every year, you have over 40 million mammograms in the US alone. So worldwide, you have hundreds of millions of mammograms done every year primarily to detect breast cancer. So of course, you can train, and people are doing that, deep learning methods on detecting cancer in images of the breast. But here we did something a little bit different, which is detecting the level of calcification in the breast, right? So you have blood vessels, and sometimes you have calcification on those blood vessels, which is visible under the X-rays. And so you can estimate what is the total level of calcification in a given image. Now it's a regression problem. So of course you need expert radiologists to measure the level of calcification in your target data to build a training set. So we have that, and we train a deep network, and you can see here that the reference, that is the human assessment of the calcification and the predicted calcification level by the neural network, you see that there is quite a good agreement. And of course, the reason for doing that is that a high level of calcification is potentially a sign of a heart disease. So as a byproduct of being screened for breast cancer, you could also be screened for risk of heart disease if you have a very high level of calcification, found to have a very high level of calcification. You could be referred to other doctors in order to do additional tests for heart disease, right? So that's a regression problem. This is another problem where the task is to look at these images, and these are images of the spine. And you want to know whether these are patients with cancer. You want to know if the cancer has metastasized to the spine, right? That's the cancer tend to metastasize first to the liver, then the lungs, and then the spine. It's the most likely, third most likely target. And so again, you can train convolutional network and identify whether there is a metastasis in the spine, and where sometimes there could be two regions of metastasis and so on. And so again, we have done this with a convolutional neural network, nothing special there. Another example is a system that we have that is capable of identifying hair follicles in images of the scalp and count hair follicles. This is important in applications, in dermatology application, where you are, say, developing a drug for promoting hair growth, for instance. The only way to really be sure that it works would be to take an area of the scalp, count how many hairs are present before the treatment, and then during the treatment and after treatment in a very precise way. And the only way you can do that is by developing these kinds of methods. We've done applications in radiological images of the brain here, detecting genetic mutations in gliomas. Won't go into the details. This is just an example of work done by other groups out of many thousands, of course. This is a group at Stanford that had an interesting application where you could take images of moles of your skin and have a deep neural network classify into different categories, whether it's malignant or not, different types of malignancy and so on. This could be very useful, for instance, especially in developing countries or rural area where you don't have immediate access to doctors. You could have take pictures of areas of your skin with your cell phone, send them to the cloud, and get some feedback immediately on your cell phone. So I could go on and on. These are all applications to images, but I'm going to show you something different now, which is applications to video. So for example, we worked on colonoscopies. Again, millions of colonoscopies are done every year. And the idea is to go inside your colon and look for polyps. Polyps are precursors to colon cancer. So they look like this, like you can see in this image. And during a colonoscopy, a doctor will remove the polyps from your colon and then send them to pathology to see whether they are malignant or not. The problem is that the area of your colon is roughly the size of a tennis court. And colonoscopy have to be done fast, something like 5 minutes, for instance. And so it's not uncommon for doctor to miss polyps. And so we wanted to build a system that can help them find all the polyps. And of course, it's a video, right? A colonoscopy is a video. And the question is, how can you adapt the methods, the CNNs, the convolutional neural networks to this problem where you're dealing with videos? So we worked with doctors that were able to give us a labeled data set of several thousand images of the colon with polyps and without polyps. So you can view it first as a classification problem. There is a polyp, so there are no polyps in the image. But then, of course, you want also, if there is a polyp, you know where it is. So you want to draw a bounding box around it. And this can be done. There are various techniques now, but one of the most widely used is the so-called YOLO technique. You look only once. But the basic idea is that the output of the neural network is not just a binary, a logistic unit that tells you yes or no, the probability that there is a polyp in the image. You can also have additional outputs that represents basically the coordinates of the bounding box. So the center of gravity and the width and the height of the bounding box could be a good example. You can take these 2 points, the XY coordinates of 2 diagonally opposite points of the bounding box. And so you can train those via regression. So you have both classification and regression units being trained at the same time. So we did that for the images, but then again, you say, well, that's nice, but you're dealing with images. You're not dealing with videos. How are you going to deal with videos? Well, it turns out that it's very easy. You don't have to do anything else, essentially, because training the network on images takes maybe a week, depending on how many GPUs you have, et cetera, But once the neural network, the CNN, is trained, it takes about 20 milliseconds to produce the output, right? So if you have 20 milliseconds, even if you have a video with say 60 frames per second, you're not going to do a prediction at every frame, but you can do a prediction, say for 10 frames within a second. You have plenty of time to do that. You can do better than that, but 10 is plenty to detect whether there is a polyp or not. And you can even use neighboring frames to make your inferences more robust, right? So if you have a frame that says there is a polyp, and then all the frames around it say there is no polyp, most likely it was a false positive, right? So that's what we did. And we now have a system that is that has been further developed and I would say even copied, and is now being used actually in the clinic. And I'm going to show you here how it operates in real time. So this is real-time predictions. You're going to see a collage of videos of colonoscopies, and you're going to see our AI system. People call it an AI system, but it's basically a CNN that puts the bounding boxes in real time around the polyps. And it's about as good as a human expert, except that it doesn't miss us anything, of course. And of course, this is used. It's used in the clinic right now, not to replace the doctors, of course, but to help the doctors. You can imagine having various interface. You could have a beeping sound or whatever to help the doctor really avoid missing any polyps. Another example I'm going to show you is this is in the eye, and in eye surgery there is something called the TM, the trabecular meshwork here, which is this line here, which is very hard to see, which is a very important marker during eye surgery. And so you would like to have a system that is capable of identifying the TM during surgery. And so I'm going to show you the system again we have developed using convolutional neural networks in real time. But again, it's a CNN that can be applied many times every second that produces these lines. To help surgeons during eye surgery. Okay, let me show you maybe a last one is an application to veterinary medicine, right? There is no reason biomedical imaging is not just humans. You can apply it to animals. Maybe you can study polar bears in Norway. If you do x-rays of polar bear, you want to detect certain things. Well, that would be a very unique data set that you could get in very few places in the world, Norway being one of them. We worked on a disease called valley fever, which is caused by a fungus, which is becoming increasingly it's a growing problem, for instance, in the United States, in the southern western states, primarily, but the area of the endemic area is growing due to global warming, right? So it's predicted that in 20 years, this disease will start extending quite far north. It's a disease that typically dogs can get because they are close to the dust, close to the soil, but it can be also, humans can get the disease and many other animals. But anyway, we studied it in dogs. And so we had x-rays. This is just plain x-rays of dogs with or without the disease. And in this case, it's back to normal standard CNN to detect the disease in the lungs of dogs. The challenge here, what is interesting in this problem is that the variability in the anatomy of dogs is the range of variability is greater than for humans, right? You have small dogs and very big dogs and so forth. And so There is quite a bit of variability, but again, you can train CNN, and we got very good ROC curves for these problems using a variety of neural networks. So I'm getting close to the end. I want to leave a few minutes for questions. So let me just say a few things. Deep learning, feed-forward neural network, recursive neural networks are very effective and flexible. It can be applied to all kinds of problems in biomedicines. CNN in particular are very effective when in biomedical imaging problems. And you have problems at all scales, from the microscopic to the population level, and all kinds of imaging modality, whether it's microscopy, X-ray, MRI, ultrasounds, and so on and so forth. The key thing is the data. Getting the data in the biomedical space sometimes can be difficult. For imaging problems, ideally, as a minimum, you would like to have a few thousand images labeled, but you can get away many times with less than that. So we've done problems where we had, 100, even less than 100 images, labeled images. And when you have little data, you need to do a number of tricks. Of course, regularization to avoid overfitting, dropout, things like dropout, etc. You can do data augmentation, that is, you create additional data by a number of tricks. If the problem says symmetric, you can flip the images, rotate the images. If it's rotation invariance, you rotate the images. You can add Gaussian noise, etc. Many ways of augmenting your data set. But probably the most useful technique for dealing with relatively sparse data sets is to use pre-trained networks. That is, you use networks that were trained for imaging problems that are completely different, like ImageNet or regular visions or self-driving cars. There are big networks, something like VGG16, for those of you who are familiar with those networks and those techniques. So you take those big networks that were trained, say, by Google or Facebook, a big company like this, on general purpose vision, and you apply to your problem. All you have to do is to retrain the top of the architecture. All the lower layers, the feature detectors, the edge detectors are already trained and good for your problem. So you just put a small thin layer at the top with not many weights, and then you train it with your actual data, right? And that works quite well for a lot of problems. There are techniques for compressing deep networks, such as so-called distillations, which can be important if your network has to be deployed on a machine that has constraints of memory, for instance. That's also possible. There are a few interesting open problems in vision still. Things like 3D convolutions. Of course, we can do 3D convolutional neural network, but they are very expensive computationally. So there are interesting questions on how you can handle 3D convolution efficiently. And potentially there are very complex image problems in some cases where you have a lot of occlusion and so forth, where plain CNN may not be able to do well, which are still interesting to work on. But for most of the basic biomedical imaging questions, I think CNN, if you have the data, CNN will do the job for you at a level comparable, if not better, to the level of doctors. There is the black box problems that I mentioned that people complain about for deep learning. My view is that it's inevitable. In a sense, it's part of the business. It's due to the fact that the storage, really the essential, the root problem is that the storage in neural network is completely different than the storage in a Turing machine, where you have a tape and things are organized in a nice way along the tape. This is completely different here. You're taking your training set and you are shattering it and pushing them into the weights of this neural network, as you do already in linear regression. And so that process that is not invertible is the root of this opacity. Nevertheless, there are a number of things you can do, trying to design explainable architecture and so forth. I think one of the main things for interpretability and dealing with the black box problem is to do encapsulation. That is to break up your problem in a lot of sub-problems and have neural network deep learning methods deal with each one of the such problems, so that then you can identify where the problems are. In case of failure, you can identify the source of the problem and fix it. A good example is in the colonoscopy problem. We were getting false positives, right? So the system was saying, oh, there is a polyp in this image, and it was wrong. And if all you have is a network that says there is a polyp and there is no polyp, you don't know what to do, right? You don't know how to fix it. But if you do also localization, that is, if you have the bounding box, now the network is telling you there is a polyp and I think it's here, and you have the box, and then you can look at what's in the box and identify what was the problem. In our case, it was the presence of surgical instruments. So every time there was a surgical instrument in the picture, in the frame, the network was predicting polyp, because there is a correlation. Of course, the instruments are usually close to the polyps, but it's not the presence of a polyp, right? So by doing this two-level thing, is there a polyp or not? And if there is a polyp, where is it, right? That's the encapsulation, the multi-level analysis of the image. Then you can detect that it's due to surgical instrument, and now you can fix it by either adding an extra output that predicts whether there is a surgical instrument or not, or changing your training set, etc., right? The main challenges in this space in biomedical imaging is getting the data, number one, and then of course pushing the technology to the clinic, because there are many issues there beyond just the technology, in terms of radiologists or other types of doctors being afraid of losing their jobs, in terms of hospitals being afraid of lawsuits. What if the neural network makes a mistake, a patient dies, etc., etc. So there are legal issues, there are privacy issues, security issues in getting the data, and so on and so forth. So lots of interesting issues that are around the area of biomedical imaging, but not really, technical questions. And of course, moving towards general AI is another, of course, central challenge, not only for biomedical imaging, but of course for the entire field. So thank you very much for your attention, and